Biology.
So hello there and welcome to this class. So today I want us to look at growth and development, but now we are looking at only the questions in growth and development topic. So let's begin with the first question. The first question is asking, define the term growth. So what is growth? That's the first question. Define the term growth. So what is growth? So simply we see that growth, this is the permanent increase in the size of an organism due to synthesizing of new organelles in the organism. So if the organism increases in size, that is growth. But why do they increase in size? So they increase in size because there is synthesis of new organelles, there is synthesis of new cells in the body of this organism, which now leads to the process which is called growth. So apart from that, the next question is asking, define the term development. So what is development? So development, you can say that this is the permanent change in complexity of the organism. That is it. If you only say that in development, then you are good to go. Permanent change in complexity of the organism. So note this, if you omit the word permanent, most likely your answer won't be correct, because in development, something must change permanently, because if maybe we had a single-celled organism which has now developed to form a multicellular organism, so that will remain permanent. This multicellular organism, maybe for example like a tree, like a human being, like a cat, like a dog, they cannot again grow back to become a single-celled organism. No, that can't be true. So that's why we are saying change in complexity. This is the permanent change in complexity. Sorry, that is development, which is the permanent change in complexity of the organism. So mind you, when you are defining growth, you must say permanent or irreversible. It's still correct. Also, when you are defining development, you must also say permanent or irreversible or non-reversible. So always make sure that you have taken note in line of definition of growth and development. So you must say permanent, or you must say non-reversible change. So the next question, we are being asked cell division. So what is cell division? Because in cell division, remember, we look at this mostly in the topic of reproduction, that is in Form 3. So in cell division, remember we said we have two types of cell divisions. If we can remind ourselves, remember we said we have mitosis, which is the cell division involving somatic cells or the body cells. Remember, that is what we said mitosis is, the cell division of body or somatic cells. After that, remember we said also we have meiosis, whereby we had two stages of meiosis, meiosis 1 and meiosis 2. But what is meiosis? So meiosis, this is the cell division which involves gametes, either the sperm in males or the ovum in females. So that is meiosis, the cell division which mainly involves gametes, which is the sperm in males and then the ovum in females. But now, this question is asking, we define cell division. So the definition of cell division, you can say, this is the splitting of a parent cell. So that splitting of a parent cell in order to form two or more daughter cells. That is the definition of cell division. So this is the splitting of the parent cell in order to obtain either two or more cells. That is what is called the cell division. And then remember, for mitosis, we get two daughter cells at the end of the stages: interphase, prophase, metaphase, anaphase and telophase. At the end, we get two daughter cells. But in meiosis, at the end, in meiosis 1, it's like mitosis, whereby we are going to only get two daughter cells. But in meiosis 2 now, the total cells we are going to get 4 daughter cells. So again, remember, in mitosis, we get two daughter cells at the end of the stage. But in meiosis, at the end of the phase, that is of the stage, we're going to get 4 daughter cells. So don't you forget, that's the definition of cell division. And please check the previous videos where we did reproduction, and so you can be well acquainted with what we studied previously. So let's go now to the next question. It's asking, what factors enable growth to occur? So what are these factors in the environment, factors within the organism that enable growth in an organism to occur? So why do organisms grow? What factors make them to increase in size? increase in shape, and etc. So the first one we are going to talk about is the availability of nutrients in the environment. So whereby we see if there are nutrients available in the environment, the organisms are going to take these nutrients. And then remember what we studied in Form 1, the topic of nutrition. We said nutrient is anything which is taken into the body to provide the body with energy. That is what we said is nutrient. So in this point we are saying availability of nutrients leads to growth, which is true. If this organism is going to take the nutrients, so this nutrient is going to be assimilated in the cells of the organism, and then after that we are going to get energy. After that the cells are going to multiply to increase. As the cells multiply and increase, remember, so the organism is also going to multiply. The organism is going to increase in size and shape. So that was one of them, availability of nutrients. Also, apart from availability of nutrients, we see availability of space. If there is adequate space in the environment, organisms are going to multiply even faster because there is enough space to accommodate the newly formed organism. So apart from that, what we have just defined in the previous question, we see that cell division and cell expansion also lead to the growth of the organism, among others. There are very many answers you can give in this, but so far we have those. So let's go to the next question, which is asking which factors are used to measure growth. So which factors are used to measure growth? Like if you look at this organism today and then you look at it the following week, so you can be able to say that this organism has grown, this organism has increased in size and shape. So what factors are used to measure growth? So how will you know that growth has taken place? That is what this question is asking. What factors are used to measure growth? So the first factor which is used to measure growth, we have mass. So you can look at you can measure the mass of the organism before and after in order to know that growth has taken place. Apart from mass, we can talk about the volume by looking at the volume of the organism before and after. You can also measure the length. Today this organism was measuring 1 centimeter. In the following week, this organism is measuring about 7 centimeters. So if you measure the length, or if you consider the length, this can also be used to measure growth. Apart from that, you can't talk about the length if you have not mentioned the height. So you can also check the height of the organism in order to determine that growth has taken place. So let's go to the next question, definition of terms. We have cell differentiation. So what is cell differentiation? So for cell differentiation, you can say that this is the process whereby young cells are modified to perform a specific function. So remember, cell differentiation, cell modification, cell specialization, what we studied in Form 1. So it is just the same definition. The modification of a cell in order to perform a specific function. That is cell differentiation or cell modification or cell specialization, what we studied back in Form 1. So remember, in this cell specialization, just to mention a few, remember we said we have a root hair cell. So this root hair cell, it was once a normal plant cell, but now this plant cell was then modified and then given a projection. So what's the function of the projection that it was given? So the function of that projection that developed is to absorb water and mineral salts. Now you see, this is a plant cell which has been modified. It was once a normal plant cell, but then it developed that projection in order to absorb water and mineral salts. So if we talk about this modification of this cell and any other cell, that is what we are referring to as cell specialization or cell differentiation or modification. So apart from that, apart from looking at the root hair cell, we also have the palisade cells which are found in the leaf, which are used to store the photosynthetic products of the plant, among others. So for the animals, we can talk about the sperm cell, whereby for the sperm cell, it was a normal animal cell. It was just a normal animal cell, but now this normal animal cell developed projection, which is now called the tail. So what is the function of this tail that developed is to now propel or to move the sperm as it flows or as it swims towards the ovum. So you see, this is also cell specialization. We once had a normal animal cell, which now developed projection, and then it was called a sperm cell. Apart from that, we also have a nerve cell, which was a normal animal cell, but it develops the dendrites, it develops the axons, it develops the myelin sheath in order to transmit and transfer impulse from one dendrite to the other dendrite. Apart from that, we have the ovum and many more. So remember, this point is asking cell specialization or differentiation. So this is the modification of a cell in order to perform a specific function. That is the definition of cell differentiation. So the other definition of term, we look at morphology. So what is morphology? So simply morphology, this is the study of the external surface or the structure of living organism. So the study of the external structure or the external surface of living organism, remember, that is what is called morphology. So like for instance, if we talk about the fur in the cat, like as you can see, if you talk about the fur in the cat, that is morphology, because we are talking about what has covered the external body part of the cat. If you talk about the skin of the humans, if you talk about we are covered with skin, if you talk about the hairs found in the dog, because the dogs are covered with hair. So if you talk about the hairs covering the dog, that is morphology, because we are studying the external surface or structure of living organism. So remember that. So morphology, kumbuka, this is the study of the external structure of living organisms, while anatomy, this is the study of now the internal structure of living organism. Morphology is external, while anatomy is the study of the internal structure of living organisms. Yeah, so the next question is asking, differentiate between growth and development. So differentiate between growth and development. So remember, in biology, there are two ways you can differentiate. You can either use a table or you can use a conjunction between point A and point B. Like for instance, let's differentiate between growth and development. So you can say growth is the permanent increase in size of the organism. Increase in size, height, length of the organism. While development, this is the change in complexity of an organism. So if the organism changes complexity, remember we said from a simple organism to a complex organism, single-celled organism to a multicellular organism, that is development. That is the definition of now the development or development. So in this differentiation, we have used a conjunction between growth and development. So whereby you say growth is the permanent increase in size, length, and height of the organism, while that's the conjunction, while development is the change in complexity of the organism. Now you see, we have used the conjunction. This is one way you can differentiate in biology. So the other way you can differentiate in biology is by using a table, whereby on the first side you say we have growth, on the other side you say we have development. And mind you, the table must be complete. If your table is not complete, you only write a plus, growth this side, development that side, you only write a plus like that, you're going to get it wrong, because that is not a procedure or a method to differentiate. So the method or procedure to differentiate, the table must be complete. You must complete the table in order now to differentiate anything in biology, and in this case we have growth and then development. So the next question is asking, differentiate between growth in plants and animals. So what's the difference between growth in plants and growth in animals? So first of all, you can see that growth in plants only happens in regions which are called the meristems. So in animals, growth takes place throughout the whole organism. So the whole organism in animal grows, while in plants, growth only occurs in regions which are called the meristems. As you can look at this diagram, we have the apical meristems, as you can see. So apart from that, we have now the lateral meristem. So the function of apical meristems is now to lead to the increase in the height of the organism. So the apical meristems found on the shoot, they increase the height of the plant going upwards, while the ones found in the root, they increase the height of the plant going downwards. The function of the lateral meristems and the intercalary is now to deal with the stem regions. So in plants, again, remember, growth only occurs in specific regions of the plant. It's not the whole plant organism which grows uniformly. No, it is only a portion of the plant which grows, and then, like after cells have developed, that part now becomes wooden, and then the other parts now begin again to continue the growth. While in animals, remember, growth takes place throughout the whole animal body. So the whole animal body grows. While in plants, remember, growth only takes place in the meristematic regions of the plant. So question next, we are being asked, explain the two types of seed dormancy. So explain the two types of seed dormancy. So first of all, remember, what is seed dormancy? So seed dormancy, this is the condition whereby a seed is unable to germinate even when favorable environmental conditions have been provided. So the seed will not germinate. The seed is unable to germinate even when temperature is optimum, light is optimum, there is everything, there is nutrient in the environment, the seed will still be unable to germinate. So these two types of seed dormancy, which ones are they? So the first type of seed dormancy, we have the absolute dormancy, and then we have relative dormancy. So these are the two types of seed dormancy. But now the question is asking, explain the two types of seed dormancy. So let's begin with the first one, which is now the absolute dormancy. So this absolute dormancy is where the seed cannot germinate even when conditions for germinations have been provided. So the seed will not be able to germinate even when every germinating conditions have been provided. It will still be unable to germinate. But what about relative dormancy? So in relative dormancy, we see that this is when a seed cannot germinate due to the absence of germinating factors such as water, air, and temperature. So relative dormancy mainly deals with things which we can find in the environment to assist this seed enable to facilitate the process of growth. So that's the difference between relative dormancy and absolute dormancy, which are the two types of dormancy that we studied in Form 3. So on to the next question. The next question is asking, state any causes of seed dormancy. So why do you think seeds will eventually become dormant? Every seed should grow. But why will some seeds not be able to grow even when every factor has been provided? So the first one, you can see that we have impermeable seed coat. So the meaning of impermeable means no water, no nutrient can be able to penetrate the seed coat in order to reach the embryo to facilitate the process of growth. So that is impermeable seed coat. So apart from that, we have immature embryo. Yes, now the seed coat is permeable, water can be able to move through the seed, but now the embryo is immature. The embryo cannot be able to develop, the embryo cannot be able to grow. So that is the other factor which leads to seed dormancy. Apart from that, we have germinating inhibitors, which mostly involve chemicals in the environment that prevent germination. If this seed has acquired this chemical, so this chemical is going to prevent it from germinating. For example, we have the abscisic acid, which is a very good germinating chemical inhibitor. So apart from that, we have inadequate germinating factors in the environment, whereby for some seeds you can see that water, air, warmth may not be able to be adequately provided in the environment, therefore the seed will be unable to germinate. So apart from that, we see that some seeds lack germinating hormones such as auxins, because we see the function of auxin is to facilitate the growth of the plant or to facilitate the growth of the part of the plant. So if some seeds are going to lack these germinating hormones, therefore it will mean that they are going to have delayed milestones. Eventually they are going to be dominant. So apart from that, we see some seeds lack germinating enzymes such as amylase in order to facilitate faster growth of the seed to become a seedling. So apart from talking about immature embryo, an embryo that is not matured. So apart from talking about immature embryo, also the embryo might be dead. So yes, like we can have a seed, the seed is mature, but now the embryo inside is dead. So if the embryo is dead, this will lead to seed dormancy. And apart from that, you can also have poor environmental conditions such as extreme temperatures, Apart from extreme, we can have also earthquakes, but that is rare. But let's talk about floods. You can have floods whereby in the terrestrials, if the terrestrial seed has been surrounded with water, so this seed is also not going to germinate because the environmental conditions are very poor for this seed to facilitate the process of germination. So the next question we are being asked, state ways of breaking seed dormancy. So which are the ways of breaking seed dormancy in order now to make these seeds that were unable to germinate to begin now the process of germination? That is what this question is asking. So what can we do to these dormant seeds in order to make them now begin the process of germination? So the first one we see, we have physical scarification. So this physical scarification mainly involves mechanically scratching the seed coat in order to allow water and the nutrients to penetrate the seed coat and into the embryo. So scarification, picture it like you are scratching. So you are scratching the seed coat gently, so in order to make it permeable so that water and air is able to enter, and then the embryo will get the nutrients to facilitate the process of germination. So that is one, which is physical scarification. So the other one, scarification, we can talk about chemical scarification. So whereby in physical we scratch it mechanically, but now in chemical scarification, we are going to use now the different chemicals in the environment in order to make the seed coat to be permeable, or make the seed coat to be permeable so that water and nutrients can be able to enter. So simply, this chemical scarification, this is the soaking the seeds in concentrated acid in order to break the seed coat. Because if the seed coat has been broken or has cracked, now nutrients can be able to penetrate through. So apart from that, we can talk about soaking, whereby this is placing the seeds in water in order to make the seed coats to be soft, so that more water, more nutrients can be able to penetrate through and go to the embryo in order to facilitate the process of growth. Of course, if the water enters through the seed, it is going to hydrolyze all the food substances which are found in the cotyledon. If this food has been hydrolyzed in the cotyledon, now this food is going to reach the embryo, and then the embryo is going to use this food in the cotyledon, and then apart from that, now the embryo will start to grow in order to form now the early plumule and the radicle, etc. So apart from that, you can also talk about heat treatment. So for the heat treatment, we can dwell on two aspects, whereby the first one is soaking the seeds in warm water for some time. Remember, you should not soak the seeds in hot water, because if you soak them in hot water, you will be cooking the seeds. But now, we soak the seeds in warm water. So if we soak the seeds in warm water, this is going to make the tester to be soft, and then after the tester being soft, we now plant the seeds, and then they are going to grow. So apart from that, the other one is gently roasting the seeds in open air in order to break the seed coat. So remember, it is not the hard roasting, but you are gently roasting the seeds for some time. Unaeka kando, roasting another, unaeka kando, in order to break the seed coat. And then when the seed coat is broken, again, remember, water is going to penetrate through, hydrolyze the food in the cotyledons. The embryo will use this hydrolyzed food and then begin to grow. So apart from that, we can also talk about exposure to light, whereby this is providing optimum wavelength for the seeds in order to facilitate the process of growth. Because remember, in Form 1 we say that we have autotrophic nutrition, whereby we said we have two types of autotrophic nutrition. Remember, the first one we said we have chemosynthesis, whereby this is where organisms use chemicals in the environment to facilitate the process of making their own food. So that is chemosynthesis, from the word chemo, chemical, synthesis, to make. And then apart from that, we also talked about photosynthesis, whereby in photosynthesis, remember we say that this is now where organisms use light in order to make the food. So in photosynthesis, remember we say that we have two stages, which is now the light stage and the dark stage. So for the light stage, we say that organisms use light in order to make their own food. Now in this point, which is exposure to light, it is still the same with the photosynthesis, whereby if we expose these seeds in optimum wavelength, we see that now these seeds are able to use now this light in order to continue facilitating the breaking down of the food in the cotyledon with water, and then to hydrolyze the food in order for the embryo to receive this food and nutrients to facilitate the process of growth. So apart from that, you can also talk about the next point, which is chemical treatment. So this chemical treatment, we can use the different hormones in the environment that we have, like for example, gibberellins, whereby in this chemical treatment, this is the applying of plant hormones such as gibberellins in order to promote, in order to bring about growth of the seeds. So that is using the chemical treatment. So apart from that, so you can also use, apart from physical treatment, we can also talk about biological treatment. So whereby this is introducing some species of bacteria to the seeds in order to destroy the seed coat, to gently destroy. So these bacteria are going to feed on the seed coat. So if these bacteria feed on the seed coat, now this seed is able to take in water. Remember the process, take in water. This water will hydrolyze the food in the cotyledon, and then this hydrolyzed food will be used by the embryo, and then the embryo is going to grow. So that is by using biological treatment. So apart from that, there are many. So apart from that, you can also talk about, like we can also wait for the embryo to mature before we plant the seeds. So, yeah, that can also be a remedy. Waiting for the embryo to mature before planting the seeds. That is another good way of breaking the seed dormancy. Apart from that, we can also use the method of stratification. So you can use stratification whereby we have two types of stratification, whereby we have the cold method stratification and the warm water stratification. But simply, we can use stratification among others. There are very many methods of breaking seed dormancy. So at least we have gone through as much, which can be able to assist us in our revision.
Biology.